Hi, today we want to talk about manganese and the importance of it for plant nutrition. This is an element that doesn't get a lot of attention because it is targeted for specific parts of the prairies. So I want to cover four main components in today's talk. The first one is the role of manganese in the plant. The second one is what factors affect availability. Number three is what does a manganese deficiency look like? And number four is how do we look at managing manganese nutrition from an agronomic perspective? So when we get into manganese, it is involved in 40 different enzymes being produced. But the area that I really like to focus in on is the importance of manganese as it comes to photosynthesis, and in particular, the splitting of the water molecule. And we'll dig into that in a minute. Other ones that are important are things such as lignin strength. We want to be sure that it accelerates seed germination. It's very important for rooting. It's important for oxidative stress, as well as important in reproduction. So it has a long list of things to do, and I wanted to highlight a couple of these in a little bit more detail. The first one, and in my opinion, the most important one, is photosynthesis. Without photosynthesis, we got nothing. And in this case here, if we don't have manganese, we got nothing. And what I mean by that is photosynthesis is the combination of CO2 plus H2O. You put those together, the plant converts that into oxygen and carbohydrates. But that water molecule in H2O needs to be split. And what controls the splitting of that molecule is manganese. So if we don't have manganese, we don't have photosynthesis happening. We don't have oxygen being produced. We also don't have carbohydrates being produced that are being converted to sugars, which is the food source for the plant. So not only do we not get enough oxygen as humans, the plant doesn't get enough food. So it's critical in this part. Now, manganese is also important for seedling vigor and seed vigor. Here's an example of some seeds that were manganese deficient. And you can see how there's not good lignin strength, the plants are kind of limp, and if we go below ground, we can see that the rooting doesn't look very good as well. Why doesn't the rooting look good? Well, lignin strength, number one. Number two, carbohydrates aren't being made. And if we're not making carbohydrates and converting them to sugars, there's no food for the root system. Above ground, the other thing that manganese is critical for is lignin strength, stalk strength. A lot of times when we think about lignin strength and stalk strength, we think about potassium. That's true, but manganese is critical. Look at this image here, how we're seeing with manganese deficiency, the plant is limp. And what this also now leads to is a limp plant that doesn't have good lignin strength. It has an impact if we put that plant under heat stress. And here's some really interesting work that was done with wheat plants, manganese sufficient plant, manganese deficient plant. When the plant was manganese deficient under long day lengths, the temperature of that leaf went up about 2 to 3 degrees Celsius. And if it goes up higher, that also means the plant's going to start losing more water and it's going to get more limp. And this is going to have a major impact on the productivity of that crop. Now, the other thing is we have to dig into the chemistry. I know a lot of people don't like to do this, but manganese, from a chemist's point of view, is really cool because it's one of the three elements that actually can change charge in the soil. It can go from a +2 to a +4, and then it can be reduced from a +4 to a +2. Why do you care about this? Well, this is very important because if it sits in the soil as a +2, that is available for plant uptake. But if it gets oxidized to a +4, it's no longer available. So even though the soil test may say there's a lot there actually may be not much there for the plant. So this becomes very critical when we start thinking about how do we manage our manganese. The other piece we need to understand in addition to the charge of the manganese and the availability is the fact that it's not mobile in the soil. So what happens, it's going to get taken up by the plant either through root interception or diffusion, meaning that placement of manganese is very critical at the time of planting. And this is also why a seed nutrition can be very important because it's there right when the plant needs it. So what factors affect manganese availability to the plant? Well, the two biggies are soil pH. We're going to talk about that. High pH reduces manganese availability, and also organic matter. As we increase the organic matter, or if we have peat-based soils or add manure, this can really impact manganese availability. Forms of nitrogen, cold temperatures, as well as crop rotation is some variables as well, but I want to dig into the first few. So this is just a simple chart that we all know about. The effect of manganese availability goes down as the pH goes up. So how does this relate to us in Western Canada? Well, here's a map of the 3 prairie provinces as we look at the pH of the soils. Green is a higher pH, orange is a lower pH. We can see the dark greens are going to be running pH of about 7.5 all the way up to 8. These are soils that are highly prone to have manganese deficiency. And then what we do is we layer on top of this probability of manganese deficiency the level of organic matter. So the higher the organic matter, the more green it is in these maps. And we can see that in around the outer side of the growing regions of Western Canada, we have a higher amount of organic matter combined with other variables that can trigger some manganese challenges. So when we overlay some of these variables and then we look at the manganese level in the soil, we can see that in the red areas is where we're challenged with manganese. And these are specific to select geographies, such as along the Red River Valley and the Interlake in Manitoba, west of the lakes in Manitoba, in the Peace Block, and along the western portion of Alberta. So there's targeted areas that manganese deficiencies can become a big issue. So that's why we're talking about it. So when we think about what is the critical level of manganese, well, if we use certain labs that use a DTPA extraction, the critical threshold level for manganese is 1. If you're at or below 1, you're probably manganese deficient. If you're using a lab that uses an acid extraction, the critical level for manganese is about 5 to 10. Now, the crop you grow also determines the probability of response. Now, all crops are quite responsive to manganese. The canolas, the pulse crops, and the soybeans have a high probability response, but the cereals, in particular the oats, as well as the corn, really respond to manganese as well. So we think about this and we look at the tissue samples. The tissue sample results we get back for the prairie provinces match very closely to the soil levels. Those same areas that we saw a few slides ago that show where it was red, where there was low manganese, we're seeing manganese deficiency, specifically in parts of Manitoba and the western part of Alberta. And this is going to have a major impact on yield. And we're going to talk about that impact on yield in a few minutes. So one of the other challenges with manganese when we think about manganese management is not only is it not mobile in the soil, it's not mobile in the plant. So this is where we have to be stacking little and often applications. And because it's also not mobile in the plant, it normally shows its deficiencies in the youngest growing parts of the plant. Here's some examples of a manganese deficiency. You're going to see in the middle of this field that the part of that crop is laying down. And the reason why that part is laying down is that got a double application manure. So a higher application manure triggered a manganese deficiency, and we saw this in this lignin strength being compromised with the plant being limp. And then when we zoom in on that plant, we start seeing classic manganese deficiencies in cereals. All these little dots all over the leaves that some people think are disease, but those little speckles is oxidative damage, free radical buildup in the plant caused by manganese deficiency. Soybeans, very similar. Here we leave a strip in the field in soybeans and it shows a classic manganese deficiency in soybeans. Everywhere else that field got treated with manganese. When we look up closely, we see this same piece where we have the youngest parts of the plant are showing this interveinal chlorosis, this yellowing of the plant, and on the underside of the leaf, on these extreme situations, we start seeing the same speckling as well, which is going to make it prone to infection or penetration from various diseases because of this poor tissue being formed. Canola looks very similar to the soybeans in that it's this interveinal yellowing on the youngest part of the plant as well. So if we have manganese deficiencies, and we talked about poor root development, poor lignin strength, this also makes it very prone to soil-borne diseases. In this case here, manganese deficiencies increases the incidence of rhizoctonia, a disease that some of us may be a little bit more common for us in the prairies and our growing areas is the take-all disease. And this is a root-borne disease that targets a crop, and a lot of times is very connected to manganese imbalances and manganese deficiency. So to show how we can address some of this from an agronomic perspective, we went to the Red River Valley a few years ago and we thought the soils may be manganese deficient. We grew some oats crops in these small plots and we can see that the check, which is on the left-hand side, looks very healthy. On the right-hand side, we put on a couple of applications of manganese because we felt from our soil test this plant should be manganese responsive. And from the biomass production here, you can see that there is a visual difference. When we did the tissue sample, what we saw is those plants were deficient in manganese. So we put on a foliar application on the one side, and 12 days after treatment, we saw that we increased the manganese concentration in the treated plot. Where we did put manganese, the manganese deficiency kept going down and down and down. Now, with some of our in-field analytical tools that we measures manganese in real time, we showed after 4, 18, and 34 days that manganese application kept that manganese level above the threshold, which means that we're not going to be manganese deficient. So what are that terms and means of production? Well, on this site, it meant a 5 bushel yield increase, which is actually maybe a little surprising because we didn't even think about manganese in this field prior to this. Now, this field is not alone. This is another part of Manitoba in the Interlake, which has got a peat-based soil, a high pH, and we can see massive manganese deficiency where we didn't treat the manganese on either side. Where we put on a foliar application manganese, it helped overcome some of those challenges. And of course, the plant didn't grow as fast, it wasn't as tall, wasn't as robust, the rooting system was smaller. And what that really meant was a significant drop in yield, up to 10 bushels an acre from that manganese deficiency. So again, when you have manganese deficiency, it can impact yield a great deal. Now, another crop that's very responsive to manganese is soybeans. And so we did this trial trying to say, can we kind of put on a little bit of manganese throughout the season, and what does that do for the plant? Well, in this soybean plot, the plant was manganese deficient from our first tissue sample. Once we put on pre-seed that had manganese in it as a seed nutrient dressing, it increased the amount of manganese in the plant. Still not to the optimum level. Then at the herbicide timing, we put on an application that had manganese in it in the relief, and we took up the manganese concentration in the leaf a little bit more. And this sort of stacking effect had a positive impact on productivity. The seed dressing that contained manganese increased yield about 7 bushels. The additional application of relief that had manganese in it increased it another 4 bushels. So a very responsive field to manganese. But this shows what happens if we don't address a nutrient. The yield reduction can be dramatic. So this is a lot of information to cover on manganese. We know it's critical for photosynthesis. We know that soil pH and organic matter really drive the availability. We also know, depending upon the conditions, manganese, even though it may test to be available to the plant, it may become unavailable as it can change charge. We know that it's not mobile in the soil. It's not mobile in the plant, so we got to think about this from a nutrient management perspective. And what the data is really showing is that little and often applications during the growing season is generally gets your best return on the investment. We can put some manganese in the soil, put some manganese on the seed. We like that as well, but the in-season management is really we're going to get our best bang for our buck. So to learn more about manganese or any of our other nutrients or products, go to our website today. Thank you.